All right. Well, thank you so much for joining us or tonight, depending on where you are in the world. For those of you who don't know me, my name is Natalie Doran. I am the Customer Support Director here at Diagnosis. Also joining us from Diagnosis today is Jeff Farmer, who is our CEO, and Anna Rosa Gelsberg and Richard Robson out of our European office. Diagnosis, for those of you who aren't aware of us, is the leading manufacturer of electrophysiology equipment, and we've been hosting a series of educational webinars over the last year. If you haven't been receiving invites to our webinars, please reach out to me at some point today, and I'll make sure that you're included in future invites. This week, we are lucky enough to have Dr. Tonia Rex as our speaker. She is at Vanderbilt University, and she has contributed very much to the field. And she's here today to talk with us about the Celeris system and how she's using advanced methods for ERG and VEP diagnostics to sort of aid in her research. So before I turn things over to Jeff and to Dr. Rex, I'm going to spend just a moment going over the format today. Please keep yourself muted during the presentation so as not to disrupt the speaker. The mic button is at the bottom of your GoToMeeting window on the left. If I hear crowd noise, I will be muting people. Please don't take it personally. The presentation will take about 45 minutes. There will be a Q&A session at the end. You are more than encouraged to use the chat bar on the right. There's a little chat bubble at the upper right-hand corner of your screen. If you have any questions about anything during the presentation, please feel free to put a question into chat, and we will answer it during the Q&A session afterwards. And with that, Jeff, can I have you take us into the presentation?
I will. Thank you, Natalie. I have just a couple of slides that a few of you who have joined us in past webinars have seen, but hopefully helpful for people joining us for the first time. Just as a quick background on some of the products before Dr. Rex goes through her research. So I'll go through that quickly. First, just as an introduction on Diagnosis, we've been around since 1984. We started the company. We have offices in Boston, just outside of Boston in the United States, also in Cambridge in the UK, in Dublin, Ireland. And we also are lucky enough to work with about 15 at least distributors around the world. So we have representation in most major countries around the world, and that continues to grow every year. Oops, let me do that. Quick introduction on our product line then for animal testing. So we do provide systems for human testing. Today we're focused just on animal testing. This is our lineup for our electrophysiology systems we offer for animals. Celeris first is the primary platform that Dr. Rex will talk about. She may mention a couple of points around the color dome lab cradle as well, which she has and has used. Celeris is focused on smaller mammal testing, so anywhere from baby mice up through large rabbits. The Color Dome Lab Cradle is our legacy platform, but we still sell it. It's still a very good system. That also works for rodents, and it can be part of a system that does larger mammal testing. So at the bottom of this page, I have listed it E3 with Color Dome. The E3 can drive any of our stimulators. So for customers doing work with larger mammals, usually we recommend a platform that has the E3 and then a range of different stimulators depending on what type of animals you're working with. Focusing in on Celeris. Celeris we launched as a product about 4.5 years ago now. We had a couple of goals for the system that I'll touch on now. First of all, make it much smaller than any system that we had developed in the past and any system that exists on the market today. So this is the entire system. There's a computer that drives the system with our software, and then the platform itself, which I'll talk a little bit more about. A couple of the key goals that we have, and I think have borne fruit for the system, was to make it easy to learn, easy to train. So we have a wide array of customers who are very expert customers and have been doing electrophysiology for years and decades. But we've also sold a lot of systems, a lot of Celerises to customers that have never done electrophysiology. And that was one of our primary goals with Celeris was to enable people who could benefit from the functional information they can measure with the system, but had never done ERGs, to be able to do it practically. Rapid setup of animals, depending on the protocol, you can test 10 to 20 mice per hour. We've seen that with many customers. And also very high performance. So we didn't want to have a trade-off where we made something small, lower cost, easier to use, but lower performance. We actually have a very high performance platform. We think in many ways higher performance than our older system. And then enabling a broad set of tests, which I'll talk about in a minute. So one of the key, if not the key design attribute that's different and patented on Celeris is the way we have integrated the stimulators and the electrodes. So the first thing to know is the stimulators are dicoptic. So there's two stimulators, one per eye. And as you can see in the picture center top, there's an electrode actually integrated just around the outside edge of the stimulator itself. And so in setting up an animal on Celeris, you just simply bring the electrode and the stimulator at the same time up to the animal's eye, just in front of the pupil, with a drop of ophthalmic gel, and that provides the electrical contact between the eye and the electrode. So it's one step to position the electrode and the stimulator at the same time. And obviously they're very small. This also enables you, compared to a Ganzfeld setup, to be able to see the animal during the testing, which can be quite helpful as well. So we have those in a range of architectures and sizes. We have full field stimulators, which are shown on the right-hand side, anywhere again from baby mice up through rabbits. This architecture is used specifically for baby mice up through rats. And then over on the left-hand side, we have a pattern stimulator. And again, this is a real key enabler for a lot of tests that people have done in the past with larger monitors, but were very, very challenging to execute. In this case, we've got the monitor that's just about 1.5 inches by 2 inches, very small, in this package that you see in the lower left-hand side, again with the integrated electrode. So that opens up pattern tests both for ERG and VEP as well. That architecture then enables most of our protocols to be done in what we call a touch-touch setup. And what we mean by that is no additional electrodes are needed other than the 2 electrodes that are placed on the eye that are part of the stimulator. So no reference electrode, no needle electrodes that are used, no needle electrodes that are needed for reference or ground for most of our protocols, not all of them. And so for those protocols, we're actively stimulating one eye, doing a recording with that electrode, and then using the fellow eye as the reference as well. That has speed advantages that are significant in terms of setting up the animal, also speed in actually doing the test, and as it turns out, creates highly repeatable tests as well. Rapid ability to customize protocols, so that comes as part of the software package that we use on Celeris as well as all of our other systems, multiple ways to export data. And then we have worked with some of our customers to customize add-ons that you can place on Celeris or with Celeris as well. I'll show just the breadth of tests. So Dr. Rex will talk specifically about a number of these, but just to give everybody a sense of all the different types of tests you can do on Celeris, this shows examples of the full field tests that are capable of being done on Celeris. These can be done across a wide range of luminances as well as colors. So the colors that we offer are UV, blue, green, and red, and you can do any combination of those in a test from below the scotopic threshold up over 1,000 candela-second per meter squared flashes. Those then enable these types of tests: dark adapted ERG, C waves, light adapted ERGs, as well as scotopic threshold responses in the bottom left-hand corner, flash VEPs, photopic negative responses, as well as I show an example here of an ERG done using the UV LED sources as well, which is obviously quite helpful for rodents. This shows some examples of pattern-based tests. So of course, pattern ERGs are very common on Celeris for our customers who decide to buy a pattern ERG or a pattern stimulator. We show here pattern ERG responses from a mouse, a rat, guinea pig, and other customers have used other animal models as well as a pattern VEP example in the bottom left-hand corner. if you decide, so Celeris is very modular. If you don't already own one, our sales team works with a prospective new customer to understand their tasks and then configures the system with the proper simulators. So if you have a pattern stimulator, both of those tests will come along with your stimulator in the system. And then we're always working on new tests as well. So I show a multifocal ERG that is in development for Celeris on its pattern stimulator also. And then finally, I won't cover all of these, but just to put this up for a couple of seconds, this is our current list of all of the standard protocols that come along with Celeris. If you buy the stimulator that enables that protocol, you will get these protocols. And then we also do a lot of work in customizing our protocols to help customers meet their specific research needs. And that's an area that we try and differentiate ourselves and just try to be helpful. If we don't have something that meets your exact needs, we'll work with you to try to do so. So with that quick introduction on Celeris, I will go to the next slide and turn things over to Dr. Rex. Oh, Tonia, you are on mute.
Thank you. Thank you, Natalie and Jeff, for the introduction and the opportunity to share about how I use the Celeris system in my research. I'm not going to go through a lot of this because Jeff just did such a nice job, but I do want to show this is one of our mice on our Celeris system. You can see we have the electrodes on the eyes, but we also have, you might be able to see a little bit right here, a subdermal electrode in the front of the animal's head in the snout region as a reference, and then we have additional electrodes in the back and then a ground back here, and that's so that we can also do simultaneous VEPs. There we go. So this is what the result looks like on the screen of the system. Just looking at a cross section of a mouse eye here, light comes through. It's going to travel through the layers of the retina, and the first electrical activity happens with phototransduction. That's seen with the activity of the photoreceptors, which is seen by the A wave here. And then we have a B wave, B max, that's from the bipolar cells. And these oscillatory potentials can be pulled out. They're shown here and here for the two different eyes. And that's driven by activity of the amacrine cells. And then finally, these two waveforms down here are from the two sides of the visual cortex. So the visually evoked potential is the N1 and the P1. And again, that's recorded from the back of the head after light stimulus. So that's recording activity from the whole visual pathway then. With the VEP and having to place electrodes on the back of the head, that electrode placement is incredibly important. Obviously, the electrodes on the eye need to be placed just right, but we know where the eye is. And finding the visual cortex can be a little trickier for researchers the first time they're setting up. So this paper by Ritter and Nisinowitz from 2006 is really nice because they show how electrode placement affects what you record after exposure to light. So here, when the electrode's on the eye, you can see we get the nice ERG waveform. And when we look back, down at E and F, you're getting the visually evoked potential here. And then you can see how that changes as you move the electrodes further above the head. So electrode placement can be really important. One thing some of our staff will do is remove some of the fur on the back of the head or even use a Sharpie to draw so that they know exactly where to place that electrode and they know it's where it belongs. I'm going to talk to you today in part on a model of glaucoma. I study glaucoma because it's one of the leading causes of blindness worldwide. And while we understand that it's age-related and there are certainly some genetic components, there's no clear genetic etiology for the vast majority of glaucoma cases. And in fact, the only modifiable risk factor is sensitivity to intraocular pressure. Current medical interventions include drops to lower that IOP, but there's poor patient compliance with those, or surgery to lower IOP. But we have the additional problem that some patients, even with lowered IOP, still progress to neurodegeneration and vision loss. And that is due to degeneration of retinal ganglion cell axons in the optic nerve and death of those cells. So there's a need for treatments that target those neurons. And here's some examples of what happens in glaucoma is this progressive loss of peripheral vision. And most patients don't realize they've lost vision until they're in the advanced stages. So that's another big challenge in glaucoma is how do we identify it earlier? So we use a mouse model. This is the microbead occlusion model where we inject microbeads into the anterior chamber. Those clog Schlemm's canal, resulting in elevated pressure within the eye and damage to the retinal ganglion cells. And here are some IOP measurements over time in saline injected animals versus the microbead injected animals. And you can see that we have sustained elevation in intraocular pressure for at least 4 weeks, and we can always re-inject as needed to go longer. This model was developed by Drs. Calkins and Sappington. This is a diagram from Dr. Calkins where he have demonstrated what tends to happen in glaucoma, where we know that there is an axonopathy that occurs, and there's also dendritic pruning that occurs within the retina. So it's important to address both those processes. Cell death is actually a very late process. So one recent finding that we had that's really exciting is that we get a deficit in the photopic negative response 2 weeks after elevation in intraocular pressure. Now, when we perform the visually evoked potential, which we collect in the same session as a photopic negative response, then at 2 weeks, there's really no difference from control. It takes 5 weeks to see a deficit by the visually evoked potential. Importantly, and here's the quantification over here, right? So we have this drop in the photopic negative, whereas the VEP is normal. Importantly, if we look at the optic nerve, the total number of axons and the number of degenerative axons is no different between our control and our glaucomatous animals. So this decrease in the photopic negative response is occurring prior to pathogenesis. So it's an early marker, which is very exciting because the photopic negative response could also be performed in the clinic. So we continued to investigate what was happening at 2 weeks in this model. And what you see here are micron 4 images just showing red fluorescence from DHE, which is a chemical that reacts to superoxide. So hopefully you can see that we have superoxide present in our hypertensive animals, but not in the saline controls, and that's quantified here. And notably, the peak in the superoxide is at 2 weeks. We also performed an oxidative stress microarray that I don't show here, and it showed increased mRNA levels of many antioxidant proteins. And so we quantified a subset of these by Western, and in fact, we do have an elevation of multiple antioxidant proteins at sometimes 1, but all of them at 2 weeks, suggesting that there might be an endogenous antioxidant response occurring at this time point as well. So we further explored that. hypothesizing that since many of these proteins are driven by the antioxidant response element, which is activated by the transcription factor NRF2, that perhaps NRF2 was being activated. So here we have a Western blot looking at total and phosphorylated NRF2, and sure enough, at 2 weeks, we see this elevation in phosphorylated NRF2. To explore how that was happening, we looked at a variety of signaling proteins, and what we found was that both AKT and PI3 kinase had increased phosphorylation at 1 and 2 weeks post elevation of intraocular pressure. Now, if we block this pathway either by inhibiting either one of these phosphorylation events, and I'm just showing one of the examples down here, then we have prevented that phosphorylation of NRF2 and the elevation in these antioxidant proteins, suggesting that this is the pathway involved in this endogenous antioxidant response. So what is it doing? Does it have any impact on glaucoma pathogenesis? To explore that, we then removed NRF2 using a knockout animal and induced ocular hypertension and explored effects on these antioxidant proteins. And in fact, if we don't have NRF2 present, we don't get that antioxidant response, this increase in antioxidant proteins. So it is mediated by NRF2. And if we then look at visual function and the structure of the optic nerve, what we see is, and these arrows are pointing towards degenerative axons in the optic nerve. This is all at 2 weeks, right? So we don't expect to see a whole lot because we don't get a lot of axon degeneration in this model until about 5 weeks. So here in our knockout animal that's ocular hypertensive, we actually do have a decrease in total axons and we have an increase in degenerative axons, suggesting that we have an earlier onset of glaucoma pathogenesis. And when we look at the VEP and the photopic negative response, similarly, we have a decrease in this visual function. So in summary for this piece of my presentation, the photopic negative response is an early biomarker of ocular hypertension. It correlates with elevated reactive oxygen species at this time point and an endogenous antioxidant response of the retina mediated by the NRF2 and ARE pathway, suggesting that this pathway contributes to the delayed onset of glaucoma pathogenesis that occurs, and it could be a potential therapeutic target, and that we might want to consider using the photopic negative response as a way to screen for glaucoma patients. It might be more sensitive and earlier than current metrics. So now I'm going to switch models to another model of optic neuropathy, and this is a neurotrauma model. Visual dysfunction occurs in about 70% of traumatic brain injury patients. Traumatic optic neuropathies can also occur, often coincident with traumatic brain injuries, and indirect traumatic optic neuropathy, or ITON, is more common than direct traumatic optic neuropathy, which is modeled by an optic nerve crush or exotomy. There are no existing treatments for ITON that are more safe or effective than observation alone. The approaches that can be taken are high-dose steroids, which we know have significant side effects, or fenestration surgery, which is quite a delicate procedure. So in my model, we direct 15 psi air blasts at one eye of the animal. Here's a schematic of the system. You can see an anesthetized animal here. It's slid into this housing chamber. There's a small hole here and just across from the machined barrel, and you can see the mouse eye right there. So the rest of the mouse is protected. This is really a very focal event. We have found that at 15 psi, one exposure is really non-injurious, but if we repeat that, we get significant injury depending on the timing between blasts. And this models the repeated injuries that occur when a linked mine explodes, or when you are in the presence of an improvised explosive device, or an IED, in which case you have the initial blast wave that can then hit surfaces near a person, rebounding and amplifying, and can also cause fragments to impact, and can cause the individual's body to make impact against a hard surface. So it's actually many traumatic events that occur in a very short time period. That's what we are modeling. So when we did this, we found when we performed a flash ERG, these are some representative ERG waveforms for our different groups here, but just looking at our sham and I2N group, the AMAX, there was no statistically significant difference, but when we looked at the B wave amplitude, there was a decrease, which was unexpected because by OCT and histology, the retina appeared normal. The latency for the Bmax was also increased, and the latency for the Amax was not, and that's not shown here. So for a variety of reasons, we decided to treat with galantamine. It's just based on some other studies and kind of mechanism we thought might be happening. So we started giving the animals galantamine in the drinking water after the last blast, which is the 3rd day, and then performed analyses 1 month after injury. And with galantamine then, we mitigated some of this decrease in the Bmax amplitude, and we preserved the latency. So let's investigate why this Bmax was decreased, right? If the Amax is normal, the photoreceptors should be fine. So we hypothesized it was something about the connection between the photoreceptors and the rod bipolar cells, because in the literature there is suggestion of what's referred to as a hidden synaptopathy, where their synapses can be disrupted. So we performed immunolabeling with synaptophysin, which labels the ribbon synapses and the rod photoreceptor terminals, and PKC alpha, which labels the rod bipolar cells. And hopefully here in the sham, you can see that there's overlap. You can see the nice dendritic trees coming up from the bipolar cells. And in our trauma model, you can see some of these dendrites are retracted and look almost spherical. And we don't have the overlap with the synaptophysin. And we even quantified this here. So in fact, we have rod bipolar cell dendrite retraction and loss of that synaptic connectivity. And when we treat with galantamine, we have preserved the synaptic connections, and that explains our preservation of the Bmax. Now, we originally had performed the ERG, again, in the same session that we did the VEP, really as a control, because the VEP measures the whole visual pathway, and I was expecting optic nerve damage and decreases in the VEP, but I had to make sure that the ERG was functioning normally. But it's a good thing I ran it, because apparently there was this change in the Bmax. But here you can see we get a very large decrease in the VEP. This is again at 1 month. So sham is the red. Our I2N group is in green here. And here you can see the quantifications. And this correlates with the histology. So these are cross sections of optic nerves. All these little circles are axons in the optic nerve with their tight myelin sheaths. And if you look at our injury group, hopefully you can appreciate this large amount of glial hypertrophy. The glia have expanded to take over the space left by the axons that died. And you can also see quite a few of these little dark spots, which are degenerative axons. And when we treat with galantamine, we preserve the VEP amplitude and the latency and the total number of axons. So here, hopefully I've convinced you that blast-induced ITON causes disruption of connectivity in the retina despite a normal retinal morphology. That we have a decreased VEP amplitude that correlates with axon degeneration and glial hypertrophy in the optic nerve. And treatment with galantamine beginning on the last of 3 days of injury was partially protective and able to mitigate these effects. All right. So now I'm going to stick with the injury model, but talk more about antioxidants and oxidative stress again. So here is that same DHE labeling I showed before with the glaucoma model. And sure enough, in our injury model, we also have an increase. This is injury and this is sham. Sorry, I apparently forgot to label those. But we've quantified it here. Oh, no. So this is matching the graph. So we've elevated DHE and blast, and this happens to also have had PLGA particles, which are the gold standard, right, in the field is PLGA to deliver drugs. And here we delivered PLGA particles carrying a modified form of erythropoietin. So it has an amino acid change that causes it to be less hematopoietic. We also quantified levels of SOD2, the mitochondrial superoxide dismutase, and unlike in glaucoma, where we saw this increase in antioxidant proteins and this endogenous protective response, here we see that after injury, we get this very large decrease in SOD2, suggesting the mitochondria are injured with a susceptibility now of the retina to oxidative damage. And here are those PLGA particles. And here's the PLGA with EPO showing some protection. What's more interesting is when we look at this group here, this PPS. So these are novel particles that break down in the presence of certain oxidative molecules. And so they break down by reacting with those ROS species, which then results in essentially a chemical antioxidant effect. So I like to say they're inherently antioxidant. And then we loaded some of them with our EPO protein. And here you can see some partial protection just with the empty particles. But when we have them also loaded with EPO, we have this really nice preservation of SOD2 levels. As expected, when you have oxidative stress, you also often have inflammation. And in our trauma model, it seems to be very specific to the IL-1 pathway. So we have this elevation in IL-1 alpha, as well as an IL-1 beta in the optic nerve. This also happens in the retina. And using the PPS particles alone or with EPO, we block that inflammatory reaction. PLGA appears to be inflammatory, but when we have EPO loaded in those particles, we still get protection. So what happens to visual function then? Because that really is what matters at the end of the day. So here are the flash ERG and flash VEP that were acquired during the same session. You can see that we have preservation of both the ERG and the VEP by treatment with our particles, and it's quantified down here. So you can see that loss in the V wave amplitude in our injury model that does not occur to nearly the same extent in our treatment groups. Same thing with the latency. And then with the VEP amplitude and latency as well, we get protection with both particles, although the best case is when also loaded with our EPO protein. So in summary, I've shown that the trauma model causes oxidative stress and activation of the IL-1 pathway. PLGA can be inflammatory, and that the PPS particles and EPO, arsine-6E, both mitigate the oxidative stress and inflammation, and most importantly, they protect against visual function loss. So those are just some examples of how I use the Celeris system in my research program. And I wanted to put this quote up from my graduate student because we were talking about the Celeris system and talking about this presentation. And she said that to her, because for me, it's being able to do the ERG and the VEP simultaneously. But for her, on top of that, she was pointing out how great it is that it's portable. It's just so easy to move around as needed. And in fact, that's a really important point for us coming up because we are starting a collaboration with a group across the country that have no expertise in visual function analyses. And we're just going to be able to pick up our Celeris system and take it with us over there and set it up in a dark room and perform that visual function for our collaborator, which is fantastic. And then her other point, she says, it's been so nice. The diagnosis is so accessible and I can reach out to them with problems that I'm having, or if I want to try out a new protocol, they just send everything over and reply very quickly. And it's so true. We've worked with Natalie, we've worked with Agatha. Everyone we've worked with has been fantastic. And we very much appreciate that. And I've just circled my graduate student here, Sarah Naguib. She is going to be defending her dissertation this fall, and she's the one who performed the bulk of this work. She's been fantastic. And I also want to thank my funding agencies, the Department of Defense and the National Eye Institute in particular. So thank you for your time and attention.
Excellent. Well done. Natalie.
Well, thank you, Tonia. That was really fantastic. We have a couple of questions for you in the chat. So first question I have, the early decrease in the photopic negative is very interesting. It looks like it might be mostly due to a decrease in the B wave amplitude potentially. Do you think you could talk a little bit more about that?
Right. So the decrease in the B wave amplitude I showed you was at 1 month. It's not present at 2 weeks. So the photopic negative response decrease is actually earlier. Great question. I didn't point that out.
Great. So this one, I'm actually going to interject another question first. You have a pattern ERG stimulator, or a pattern stimulator, I suppose, but you do not currently use it. Is that correct?
It is correct.
Okay. Because I've got a couple of questions about that. Somebody asked, have you tried the pattern ERG to detect damage to the RGC and RGC axons in the glaucoma and traumatic models? So you have not actually had a chance to try that. You've just mostly been working with the flash VEP. Is that correct?
Right. So we were working on the pattern ERG simultaneously with Bruce and the development by diagnosis. And then when some bumps were hit, but we needed the information about how the ganglion cells were working, we decided that while you guys optimized and got it up and running, we were going to try the photopic negative. So we had that up and running first, and then obviously if you want to submit a paper, it all has to be consistent. So we've just stuck with it, but we will probably, revisit the pattern ERG soon.
Just a bit of context to that, to put a number around it. Tonia, I think you were one of our first 10 users of Celeris, actually, and the pattern stimulator was developed after the initial launch. So
Yes, I think I was one of the
first.
first.
We just missed the timing for you. Yeah, yep, you're right.
Yeah, I think you had one of the very first three, certainly, possibly first two. So yeah. So not going to be able to talk terribly much about pattern VEP and pattern ERG. We did have a question asking about discussing the correct placement of markers in pattern VEP and pattern ERG traces, as well as commenting on how to ID good traces. How can you tell if you've got something that's reproducible, that's legitimate, or if you've got a problem where the animal isn't seeing the stimulus properly, or you've got it contaminated with some sort of biological or electrical noise. Can you talk a little bit about that?
Yeah, absolutely. So I may be a little paranoid or old school, but I tend to export all the data. And the nice thing about the system, you export it right into Excel, and you can manipulate it from there. So I can see all the data points. I can see what the waveforms look like for each and every flash. So for the ERG, if we're doing 15 flashes, I can see all 15 waveforms. I can make sure that there wasn't something funky that happened in the middle, anywhere along the way, especially if something that might be reproducible, systematic problem. And I can also make sure that the markers are where they need to be, right? And you can just sort based on, and look at your waveform and know where to go in the Excel document. And so that's what we do. I double check my markers, make sure they're exactly where I need them to be if I'm wanting to go based on highest amplitude. There's a whole nother school of thought that you need to pick your time, and you should always measure at that particular time. Either way, I think it's fine as long as it's clear in your methods and you're consistent. But we do double check that the markers are where they need to be, especially if I get, like the V wave, right? That decrease, I didn't expect it. So the first thing I did is comb through the data and make sure that the markers were where they needed to be, that everything was right. So, I think generally speaking, the markers, you can set them in the software, they come up, and you'll get your numbers very quickly. It's very easy and very nice. But if you want to double check it, which I think is always a good thing to do, at least periodically, it's easily done with the system.
I have another question regarding the air pulse you use to generate the TBI models. Can you talk a little bit about the air pulse, like how long that blast was and how many times it was repeated?
Right, great question. The duration is a few milliseconds. This is a modified paintball marker. It's one of the only ones where you can regulate the air inflow. So that allows us to regulate how much comes out. And that's why we can set it to 15 PSI or change it as needed. Before I put the animal in, there's a hole on the other side of the housing chamber where I can slide a pressure transducer in, and that's how we measure how much air is hitting where the eye will be. Hopefully that's answered your question.
Okay. I have another one. So Ellen Saul asked, this is one for me, and actually I'm going to field it to Richard, who I understand is present. It would be nice to have a noise stimuli, as we've discussed in the past. So he's looking for stimulus that's random. So Alan, that would be a user-defined file. And what I think I'm going to do is I'm going to hook you up with Richard Robson, who's our software developer, who's a little bit more capable of building those than I am. So I'll introduce you to him. I got another question. How is scotopic measurement performed without disturbing the dark adaptation of the animal?
I mean, we just do everything in the dark. So the animals are in a dark box, and then before we take them out, the room lights are closed. We have a light tight door, and we have little head lamps with red lights, and everything's done in the dark.
The other thing to add, Natalie, would just be interstimulus interval, if you want to touch on that.
Yep, you beat me to it. So another thing you can do is basically when you're doing dark adapted testing in animals, when you present a flash, when you present a stimulus, you wait between flashes so that you don't start accidentally light adapting them. And the brighter the flash, the longer you generally want to wait. And that you can easily tell if you're starting to light adapt them because their responses will get smaller and smaller and smaller and smaller. So it's not difficult to play around with interstimulus intervals
yeah, and actually that's a great point. So software is very user friendly. So if you have some kind of genetically modified animal that needs a longer time frame to dark adapt, then you can increase that interval.
And it goes back, Tonia, to something you mentioned a few minutes ago, which is our software records all of the sweeps, each of the recordings, and so you can do benchmark tests with a model like you just mentioned to see if you've got stability in response or not, and then do what you just said. And happy to help customers. Some just do that on their own, and we're happy to help with that as well, to make adjustments.
Yeah. I mean, certainly we did when we first started working with it.
Yeah. Yep.
So another question, how do you control for the signal to noise ratio when recording? Do you have any particular tips for excluding, excuse me, electrical noise? I'll jump in after you talk and add to that, but just in your lab, how do you handle making sure that you've got a technically good test?
Right. So it's a little different between the color dome and the Celeris. With the Celeris, the way the electrodes are made, and just having everything integrated right there has ended up with a quieter recording. The color dome, there's more to have to think about and adjust. If our recordings are noisy at this point, it's usually an animal issue. The anesthesia might not be sufficiently effective, and you can see when their whiskers twitch, or something's moving. Usually it's a whisker twitch, and you can see it in the recording. So that's, you just give them a little boost, and then they're a little quieter, and then you can get your nice recording. For the colored dome, there's more external things to think about.
And so in terms of excluding electrical noise, generally the first thing you want to do is you want to make sure any power cables are located away from the Celeris, particularly from the end where the inputs are, where you plug the animal in. The other thing is, if you do wind up in an environment that has a lot of electrical noise, going through a process of elimination, it's probably some piece of equipment or electrically active thing in the room or in a nearby room, and going through and unplugging equipment that's not currently in use and won't be damaged by being unplugged can help. There's often, if you have lights on a dimmer switch, dimmer switches can often cause electrical noise. And generally some equipment is better shielded than others. We once redesigned our amplifiers totally based on some feedback from a site that had just really bad noise. And after weeks of investigating, we finally traced it back to this thing. It was a lamp. It was a lamp with a terrible cord that was just bleeding lots of noise into the room. And as soon as we unplugged it, it just all went away. If you are located in a room near large machinery, like super cold freezers or elevator machinery or the cage washer, trying to get further away from that, even if it's on the other side of the room, can help. Just distancing yourself from large machinery that uses pumps and freezers can help. And if you still have noise after all that, give us a call and we'll help you. That's my recommendation. Tonia, you talked about something there that I'd like you to go into a little bit more depth. How can you tell when an animal is on the right plane of anesthesia? How do you determine that an animal is sufficiently down for testing?
We have now done so many ERGs that we have really great historical records. I know what a good ERG should look like, and I know what those amplitudes should be. So we can use that as our gold standard and actually do this whenever we get a new staff member and we're training them, and they have to, we get them until they can do this. So if I see noise, typically that's anesthesia, especially when it's, you can see the heartbeat, or you can see those whisker movements. Those are, especially the heartbeat, obviously it's gonna be very regular, very easy to pick out. So then we know they need a little bit more. We try to only give them enough to get them at that plane. And so the chances of them being too low, we try to avoid it by minimizing how much we give them. However, if we have a low amplitude, especially in a control animal, and we've done all our checks in terms of placement of electrodes, we can even swap electrodes. Sometimes if the electrode, particularly for the VEPs, the subdermal electrodes, we clean them after every use, we sonicate them and clean them, but over time they can become dull, and that could cause the decreased amplitude and have nothing to do with anesthesia. So we just check all of those things. So, moving them around, trying new electrodes, and if none of that fixes it, then it was probably anesthesia, and this is why we do, up to 12 animals, right, to make sure that we're taking care of any variability.
So speaking of the VEP electrodes, we've had a couple of requests to look back. Jeff, can you move us back to slide 12?
Sure. Give me a second.
There we go. So we had some requests to take a closer look at the placement of the VEP electrodes. This is a common question for us. So you have the ground electrode in the hindquarters near the tail. You've got the active electrode that's placed over the visual cortex, and the reference electrode that's in the snout. Is this generally like how you place it?
Yeah.
Yeah. All right. Cool. So let's see, Jeff, you might want to put your mouse into the laser pointer. Yeah, pointer options. There you go. Just
there.
Highlight. Yeah, where that's placed.
Can you see it now, Natalie?
Yeah. Yeah.
Yep, so there's ground right here. Also, some people will put that in the tail, but hindquarter is another common area. Yep. Very common, active. And then the reference electrode, I think, Tonia, you can correct me if I'm wrong, we're going in right up here.
Yeah, I think it's laying, the needle itself is in the snout, it's just the bright part is laying on the head, right?
Yeah, yep.
I got a question. This one's for me. How do I know our Celeris protocols are the most updated version? The best way to know that is to give me your email address, and I will send you the most recent slate of protocols, or we can talk about what you've got. We're constantly refining them, but we're rarely making really big changes to our protocols. So feel free to, if you'd like to know if you're running the most recent versions, I can tell you. I can tell you how to look that up on yours. Got two more questions here. How can you validate the photopic negative protocol? Can you do some sort of pilot study where you can distinguish between an animal which has an impaired response, just to make sure the protocol will work in potentially affected animals? So if basically, all right, so can you talk a little bit about the testing that you've done about what the photopic negative looks like in these animals prior to the damage being inflicted, and that response you're seeing? What kind of differences are you seeing?
Do I still have control over the slides?
You do.
You do. Yep.
Jeff just wins in a fight. That's how it works.
I'll stay out of it.
Yeah, hands up.
There's
yeah.
the waveforms of the photopic negative response. The solid lines are the saline injected animals, which should be completely normal. And then the dotted lines are those that have elevated intraocular pressure. And the top graph is showing at 2 weeks, and the bottom graph is showing at 5 weeks.
Wow.
So, you know, we compare to our controls, I guess, is the best I can answer. Glaucoma is known at early stages, at least, to really only affect the ganglion cells. So, so I think this is a model where we're looking at the photopic negative response in a model that affects the ganglion cells, and we're seeing a deficit. You could certainly take additional wild type animals and maybe other models that are known to damage the ganglion cells specifically, right, and compare again. I don't know if that entirely answers the question.
I think so. We have a procedural question for you, a practical question. 10 to 12 animals an hour is a lot of animals. So it takes a little while to knock them down. You mentioned this last night, but not today. You use ketamine, xylazine. So anesthesia and pupil dilation have to be performed simultaneously in a number of animals, and electrode placement has to be done fairly quickly. Can you talk a little bit about, if you're doing 12 animals in an hour, what that throughput looks like?
Yeah. I will say, I don't set a timer with my staff and say you have to get through 12 animals in 1 hour. There's no requirement like that. The fact, so when there are a lot of animals to be done, different people handle things differently in the lab, but certainly we have a warming pad in the dark room, so we can have more than one animal anesthetized at a time, and they can stay warm, and we know they're safe. It doesn't take an hour to perform this procedure, so they will stay down for as long as needed to be able to record from them. Again, dilation drops can be on them while we are recording from another animal. The awesome thing with the Celeris versus the color dome is because the stimulator is in the recording, right, on these electrodes on the eye, it means, as opposed to the color dome, right, where light is filling that dome and it seeps out. And so then I worry about how well my animals are dark adapted if they're sitting out. I don't have to worry about that with the Celeris. It's small. I can have it on the whole other side of the room. The light is there right next to the eye, so you're not getting flooding of light around the animal. So we can have a staging area of prepping animals that then come through and then go back to recovery. So it's a really smooth process. My students and my staff have it down pat, and it works beautifully for them. They really like it.
One thing I'd like to add about the Celeris electrode placement is that the design of the electrodes has eliminated a lot of the variability inherent to electrode placement. Since the electrode itself is a ring that's approximately the size of the animal's eye, you're not running into the same problems that you are with gold wire electrodes, where you want to get them as close to the center of the cornea as possible without blocking light coming into the pupil. So with having the electrode be a ring that's approximately a little smaller than approximately the diameter of the eye, it doesn't matter the exact angle that you've got the electrode touching the eye with, because the average distance from every point on that ring is always going to be the same for the center of the cornea. So electrode placement doesn't take as long as it did with other types of ERG equipment.
It doesn't take as long, and you get, and it's not as finicky.
Yeah, it's eliminated a lot of that variability. So it's easier and it's easier.
Yes.
So we're pretty much right at the hour. I have one more question. If we're not going to be able to get to all of them, so feel free to follow up with us after the fact if you have any other questions that we didn't get to. We just had one comment that I want to toss to you because we talked about it last night. Do you have any comments on isoflurane or other gas anesthetics versus ketamine, xylazine?
Right. There are, gosh, and I posted it in the comments last night. I
can pull it up.
Got it? Fantastic. So I actually had this question from our IA cook. They wanted me to justify the anesthesia mix that I use. And I told them the only way I would do it is if I could publish a paper. So we worked together on it, and now there's a paper out. But in this paper, I reference a number of articles that have looked at the effects of anesthesias on the waveforms. So yes, you could use isoflurane, but your waveform's going to look different. You can also use a variety of other things. You don't have to just use ketamine, xylazine. I throw in a little bit of urethane to keep my ketamine low to allow for a greater amplitude in the ERGs. Whatever mix you use, be consistent with, because it does affect your waveform.
All right, and then final question, do you have any comments on 2-hour versus overnight dark adaptation for scotopic ERG testing?
Did you say 2-hour?
Yes.
That's a little controversial, right? So I was originally trained on ERGs by Arkady Lybarsky and Ed Pugh. And they are experts on this. And they, they look at the actual bleaching of rhodopsin, and they look at how long it takes to dark adapt, and they do the math. And really, I think 4 hours would be the shortest I would go with confidence. Because of that, and because my graduate student does not want to work through the night like a vampire, we find it makes more sense to just dark adapt overnight. The other concern with just about anything, if we're studying animals, is to always think about the circadian rhythm and how that might affect their biology and physiology. So we also like to just perform all of our ERGs, all our phys at the same, either in the morning or in the afternoon, just be consistent. So since we do a dark adaptation, we just do them all in the morning. And that way we don't have to worry about any variability that the circadian clock could introduce into any of our assessments.
I think that's what most of the customers that I'm familiar with do, Tonia, just add. And then the final point would be, of course, you can always do benchmark testing with your model, because there certainly are models that take longer to dark adapt. And the system is repeatable enough where the benchmarking doesn't take too long to do. So it's a smart thing to do.
Yeah, very true.
I'm going to give you guys the hook, Natalie. This is really good, but we're a little over the hour. So let's wrap it.
All right. Well, Tonia, thank you again. That was incredibly informative. Great talk. Really. And thank you so much for attending, guys. Great to see everyone.
Thank you.
And before we leave, I'd like to thank Tonia for a brilliant presentation. That was great, Tonia. Thank you so much.
Thank you, Bruce. Thank you for allowing me to be able to do this. I appreciate it.
It was a delight. I learned a lot from your lecture, and I'm sure I speak for all of us at Diagnosis in being very proud that we could enable research of that quality. Thank you so much.
Thank you.
Oh, and final comment. I did put the paper that Tonia referred to in the chat, so there's a link to that paper if you're interested. And with that,
thanks everyone for joining.
Thanks so much.
Thank you. Bye.
Thanks, folks.
Bye-bye.
Bye.
I don't know.